Welcome to Biology at Priyajit, my dear viewers. Today we will explore one of the most fundamental processes of life, cell division in eukaryotes. Mitosis and meiosis. Just as a building grows by adding new bricks, living organisms grow, repair and reproduce by forming new cells. This is made possible through two special types of cell division: mitosis, which helps in growth and repair, and meiosis, which produces gametes and ensure genetic variation. Understanding their concepts, stages and biological significance will help you clearly visualize how life continues from one generation to the next in a well organized and regulated manner. Mitosis, basic processes and their significance. The word mitosis comes from the Greek word mitos, meaning thread, given by Walter Fleming in 1882 because chromosomes appear like threads during cell division. Mitosis has two main parts: karyokinesis, that is division of the nucleus, cytokinesis, division of the cytoplasm, and karyokinesis occurs in five stages like prophase, prometaphase, metaphase, anaphase, and telophase. This is five subsecutive stages of the mitotic process. So what happens during prophase? Chromosomes become shorter, thick and visible due to condensation. Each chromosome has two sister chromatids joined at the centromere by cohesin protein. Condensin proteins help in chromosome compaction by supercoiling DNA. Centrosomes then separate and move to opposite poles. Spindle fibers start forming. Asters appear around centrosomes. Nuclear membrane, Golgi and ER begin to break down due to the action of cyclin B and CDK1. So mitochondria, lysosome and chloroplast remain intact. This is the image showing the different features of prophage. Now next stage is prometaphage. Nuclear envelope completely disappears at this stage. Spindle fibers attach to chromosomes at the kinetochore region. Chromosomes show jerky movements called congregation. Finally, all chromosomes move to the equatorial plate or metaphage plate. This is usually the longest stage of mitosis. The image showing the stages of prometaphage. Now, following prometaphage, the stage is metaphage, where all chromosomes align at the equatorial plane or metaphage plate. Spindle fibers are fully formed and attached to centromeres. The stage can last for different times in different cells. If spindle fibers are destroyed by cold drugs like colchicine, chromosomes scatter and the cells gets arrested in metaphage. During metaphage, APC or anaphase promoting complex destroys securin, releasing separase. Separase enzyme cuts cohesion and allowing sister chromatids to separate. Anaphase. Anaphase begins when sister chromatids separate and become daughter chromosome. It is an irreversible stage of mitosis. Chromosomes move to opposite poles by means of two movements. Anaphase A, that is chromosomes move towards spindle poles, and anaphase B, spindle poles move further apart from each other. So, and spindle fiber starts disassembling. So this is the image of anaphase telophage. In this phase, chromosomes reach the poles and decondense. Nuclear envelope reforms around each group of chromosomes. Cyclin B CDK1 becomes inactive, marking the end of mitosis. Two daughter nuclei are formed and the cell is now ready for cytokinesis. So this is the image showing the telophage of the cell division, cell cycle. Now after nuclear division, there should be a cytokinesis or cytoplasmic division. After telophage, the cytoplasm divides by a process called cytokinesis. In case of animal cells, division occurs by means of cleavage. A contractile ring is made up of actin and myosin forms around the cell. Its contraction pinches the cell into two daughter cells. But in case of plant cells, a cell plate forms at the center. This is the cell plate. Golgi vesicles collect and fuse to form a structure called phragmoplast. The cell plate grows outward and develops into a new cell wall separating the two cells. Significance of mitosis: it helps a zygote grow into a complete adult organism responsible for normal growth and cell division and development of the body. In plants, it forms new parts like roots, stems, leaves, and helps in repair. It helps in asexual or vegetative reproduction like in crops like potato, sugarcane, etc. It produces identical cells, thus maintaining genetic stability. In case of animals, it replaces worn out cells like skin, gut lining, and blood cells. Now we should go for meiosis, cell division, the basic process and significance. The term meiosis was given by Farmer and Murray in 1905. Meiosis is a special type of cell division. It reduces the chromosome number to half, that it will responsible for diploid cell into convert into haploid cell. Only one round of DNA replication occurs. Two successive divisions take place. First one is meiosis 1, reductional division, and secondly, meiosis 2 or equational division. As a result, four haploid daughter cells are formed. Meiosis 1, it consists of four main stages: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 is the longest and most complex stage. It is divided into five substages: leptotin, zygotin, packaging, diplotin, and diakinesis. Then leptotin, first substage of prophase 1, chromosomes start condensing and become visible under the microscope. Each microscope appears as a long, uh, thin thread. Although each chromosome has two sister chromatids, they are not clearly distinguishable at this stage. The ends of chromosomes attach to the inner nuclear membrane at specific points called attachment plug. Chromosomes ends gather toward one side of the nucleus, forming a bouquet arrangement. The bouquet stage helps in pairing or synapses of homologous chromosomes. So that is the leptotin, zygotin, packaging, diplotin, and diakinesis. Now we'll go for the next stage, zygotin. Zygotin is the second stage. The homologous chromosome starts to pair with each other at this stage. This pairing is called synapsis. Synapsis is performed by a protein structure called synaptonymal complex between the paired chromosomes. Each pair of homologous chromosomes now is called as bivalent. This is bivalent structure because it has two chromosomes. Since each chromosome has two chromatids, the bivalent contains four chromatids and is called a tetrad. So this is the tetrad structure. Now we will go for the packaging. Third substage of prophase 1. Synapsis is now completed. Each bivalent is clearly visible as a tetrad. Crossing over occurs between non-sister chromatids of homologous chromosomes. This involves exchange of genetic material, and crossing over mainly occurs in euchromatin regions called recombination hotspots. This process creates genetic variations. Diplotin, the synaptonymal complex then disappears. Homologous chromosomes begin to separate from each other. However, they remain attached at points of crossing over called as chiasmata, singular chiasma. The chiasmata then start moving towards the end of the chromosomes, and this movement is called terminalization. This is the image showing terminalization during diplotin phase. And diakinase is the fifth and last substage of prophase 1. Chromosomes become shorter, thick, and highly condensed. Chiasmata move completely towards the terminal ends. The image showing the terminalization process. The nuclear membrane breaks down, the nucleolus disappear, and each bivalent is clearly seen to consist of four chromatids or tetrad stage. Now, metaphase 1, bivalents or paired homologous chromosomes arrange themselves on the equatorial plate. Spindle fibers attach to the centromeres of homologous chromosomes. Orientation of each bivalent is now random and leading to independent assortment. So what happens during prophase, after prophase 1, there is metaphase, anaphase, and telophase. What happens in anaphase? And during anaphase, homologous chromosomes separate from each other. One chromosome from each pair moves to opposite poles. Sister chromatids remain attached at the centromere, and this separation is regulated by a protein called REC8. Telophase 1 and cytokinesis. This is telophase 1 and cytokinesis, the last phase. Chromosomes reach the opposite poles. A nuclear membrane may reform. Cytokinesis divides the cell into two. Two haploid daughter cells are formed. These are the two haploid daughter cells. Each chromosome still consists of two sister chromatids. Now meiosis 2. there is no DNA replication occurs between meiosis 1 and meiosis 2. The short resting phase between the meiosis 1 and meiosis 2 is called as interkinesis. Meiosis 2 is similar to mitosis and is called equational division. Prophase 2, during prophase 2 chromosomes condense again. Nuclear membrane then disappears if they are present. Spindle fibers are formed. Metaphase 2, chromosomes align at the equatorial plane, then spindle fibers attach to the centromere. What happens in anaphase 2? Centromeres divide, sister chromatids separate and move to opposite poles. This is anaphase 2. Telophase 2 and cytokinesis. Chromatids reach the poles and decondense. Nuclear membrane reforms. Again it will start to form the nuclear membrane. Cytokinesis results in the formation of 4 haploid cells. So all 4 cells are genetically different due to crossing over and independent assortment. Types of meiosis. There are basically 3 types of meiosis. Gametic meiosis, which occurs in animal cells, takes place during gamete formation. It is seen in organisms with diplontic life cycle. Second one is zygotic meiosis, which found in algae and fungi. It takes place in the zygote, found in haplontic life cycle. And third one is the sporic meiosis or intermediate meiosis. This happens in plants. Meiosis occurs during spore formation, seen in diplohaplontic life cycle. So what is the significance of meiosis? It maintains constant chromosome number in a species, reduces diploid number to haploid in gametes, prevents doubling of chromosomes in each generation, produces genetic variation through crossing over and independent assortment. It also forms genetically different gametes, provides new raw material for evolution and adaptation. So as we conclude, remember that mitosis and meiosis are the two fundamental modes of eukaryotic cell division that ensure growth, repair, reproduction and genetic continuity. Understanding their stages and significance help you appreciate how cells maintain stability while also generating variation. Keep revising and exploring the fascinating processes that sustain life. Thank you.